I'm very pleased to welcome you all here to introduce our guest speaker today, Dr. Carol Previs, who will speak on the oncogenic activities of p53 and Mdm2. So today's lecture is actually the Levine Lecture. So this is about the 11th to 12th Levine Lecture. It was established in 1999 from Sylvia and her late husband, Paul Levine, who made a very generous gift to establish the Levine Lecture, which is given annually. And the purpose of this lecture is to further excellence in cancer research and education of the next generations of physicians and scientists. And the Levines have been long-time supporters of Dana-Farber, particularly for women's cancer research, but also in all areas of cancer research. Sylvia is currently a member of the Susan F. Smith Center for Women's Cancer's Executive Council, playing a very active part in shepherding Dana-Farber's mission. And we're grateful to Sylvia and her family for all they've done for Dana-Farber over the years, and we're very honored to have this annual seminar, which bears this name. And I'll present Dr. Previs with a plaque with her name on it in the Levine Family Lecture. So Dr. Previs is a professor of biological sciences at Columbia University and was educated at McGill, did a postdoc at Albert Einstein and at the Weizmann Institute, where she actually started her work on SV40. And all good things started from SV40, including p53. And I first met Carol several years ago when I was a postdoc with David Livingston, and we'd go to the DNA tumor viruses at Cold Spring Harbor and Cambridge, England. Really fantastic times. And Carol, like many others, actually used the insights that SV40 gave us and branched out and entered into this p53 field and discovered many fundamental principles about p53. And p53 field is, if you know anything, is as controversial and as interesting, as far-flung as you could possibly imagine. And Dr. Previs has been at the top of that field, leading it, pointed in different directions all along the way. Really quite a remarkable career, training a number of remarkable scientists, including our own Wilfried Pastor, one of our newest faculty members here at Dana-Farber. And she's worked on p53 and family members p63, 73, as well as Mdm2 and Mdmx or Mdm4. She served as chair of study sections at the NIH in experimental virology and the cellular micropathology study sections, which we know is incredibly important work for all of us. Has been on the Intramural Scientific Advisory Committee for the NCI, and has been on our Dana-Farber's advisory board, as well as the Mass General Advisory Board, as well as Sloan Kettering. She's been on the advisory board of AACR as well. And you might know her as scientific editors at Cell, Molecular Cell, and Genes Development, and PNAS, where she takes particularly interest in papers that are addressing p53 and Mdm2. She is a member of the American Cancer Society, AACR, National Academy of Science, National Academy of Medicine, the Royal Society, and she's an American Cancer Society research professor. She's had many named lectures, including now the Levine Family Lecture. So we welcome Dr. Previs here.
Thank you.
Enjoy
Very
yourself.
nice. Really nice. Thank you very much. Hope I don't forget it. Thank you very much for that sort of embarrassing introduction. It's really nice to be here. I've had a wonderful day and a wonderful evening, and it's just great to hear the great science of all the people that I've met, and I'm very happy to be here. So I'm going to tell you three little stories that are built on other work that has been done in the field. Whoops, what does this thing say? Dismiss. Okay. All right. So I work on p53. I don't think I have to spend a huge amount of time describing it to you. It's clearly a major tumor suppressor. It The two best genetic pieces of evidence for that come from humans, where we know that Lee-Fermani patients harbor germline p53 mutations, and of course, your own wonderful Judy Garber has done some spectacular work, both research-wise and caring for these families. We also know that if you knock p53 out of a mouse, it'll develop cancer with 100% frequency. It was first discovered, as Jim described, in SV40, but we know that many tumor viruses have evolved to abrogate p53 function, and then there's evidence that you can actually experimentally get wild-type p53 to suppress cancer caused by other oncogenes. So we also know that p53 sits at a hub, and I'll describe a more complex hub in a moment, that involves responding to many different stresses, and these stresses themselves comprise many different types of, and these stresses produce many different kinds of outcomes, from arrest to metabolism, development, cell death, and so on. It's a very complicated protein. And the more we learn about p53, the more complicated it becomes, which is a bit daunting. We now know, for example, and I don't know if I'll allude to this again, is that the transcriptome, p53 is a transcription factor. You can see it sitting there, 4 little blue eggs sitting on a piece of DNA. That means it's a transcription factor. We all know that the p53 transcriptome varies enormously with the tissue in which it resides. So if you activate p53, for example, in liver or spleen, you'll get a completely different set of targets, both up and down. Also, there's a lot of controversy in the p53 field, and I'll probably spew out a couple of these. One of them is whether p53 is really a repressor or not, and I'm starting to believe that it is, as well as an activator. So the other thing we know about p53 is that it's a very potent protein. If it's overexpressed, it can kill or arrest cells, and so it has to be kept in check until it's needed, until the various stress signals impinge on it. And Mdm2 and Mdmx are playing primary roles in this. And we know from mice is that, for example, if you knock out Mdm2, it will cause severe embryonic lethality unless there's no p53 present. That's been also tested in many tissues in mice. And it's believed, therefore, that if it weren't for the Mdm2 and Mdmx in our cells, the p53 in our cells would kill us. So we should be happy for these two proteins. They are also, and I believe, functionally interesting in their own right. And at the end of the seminar, I'll describe why, I think. So a little more on that. As I said, it sits at the hub of many different things. And we now know, as I said, it's very complicated. And from Kastenhuber and Lowe and Cell a few years ago, they produced this very complex picture of this very complex protein, which if I spent the entire time talking about it, that would be the end of the seminar. So it's just, it's very complicated. It's also very popular to study because it's the most frequently mutated gene in cancers. Every time people find different ways to ask this question, they get the same answer. It looms over the other well-studied famous oncogenes and tumor suppressors in the frequency in which it's mutated. Its mutational spectrum is also quite unique for a tumor suppressor in that, can you see me? Yes. In that there are a set of what we call hotspot mutations that loom above the others. They might comprise between 45% of all the mutations in p53. This is sort of an old picture, and these black lines refer to essentially missense mutations. We know, however, that in many cancers, whereas about 80% of the mutations in p53 are missense, about 20% are more like truncation mutations, which means they would be more like a null mutation. So although p53 is quite unique in that many tumor suppressors are much more likely to be null when they're mutated, p53 has this rather unusual characteristic of having a lot of missense mutations, including ones with very high frequency, which has led people to believe that there are different ways in which these mutations might impact p53. So p53 is also a tetramer, and I'll talk a bit more about that in a moment. And so if you have any kind of a multimer, and most transcription factors are in fact function as multimers, dimers or tetramers and so on. If you have a mixture of a mixed tetramer, let's say, of a wild type and a mutant, the mutant is actually going to dominate, in a way, the wild type function, and we refer to this as a dominant negative. Simply having a mutation of any sort, losing p53, you're losing the wild type function. And then there is something called the gain of function. And this is where it is believed that some mutations, and there's been studies particularly on the hotspot mutations, where they actually acquire novel, what we call neomorphic properties. I have to say, I'm a big proponent of this, and I believe it, because you tend to believe your own data. But it is quite controversial. There have been some papers that have argued that there isn't gain of function. So this is a kind of discussion we can have. So the first part that I'm going to describe is the work of Josh Cho, who's sitting in the audience right here. And he did this work actually as an undergraduate at Columbia. And a wonderful visiting scientist, Tatsu Kawase, they worked together. The work was largely done by Josh, but Tatsu did some very important experiments at the end. And he was originally mentored by a wonderful postdoc in the lab, Hand Katz. And it started really with me going to a Lee Fermani symposium and meeting a patient there who wanted to offer if there was some way he could provide his own cells that could be studied. And when we found out what his mutation was, it was fascinating because it wasn't one of these hotspot mutations, but it happened to be relevant to a project that Hencast was working on because she was studying p53 as a dimer. I should say as well, many other people in the lab played a role, and David Tong, who tragically passed away a couple of years, made very important cell lines for both this project and other projects. In fact, almost every cell line in which we have CRISPR or base-edited p53 were created by David. We had a very good collaboration with Dongfang Li. I'll describe this in a moment. And of course, two physicians that helped secure cells from these patients, and Dr. Yan Zhu was very helpful as well. So what we know, as I said, p53 is a tetramer. I should say a lot of these are Josh's slides, so from when he gave a talk on this work at AACR a couple of years ago. And, and so we know it exists largely as a tetramer, and we know from Galit Lahav's beautiful work is that normally in cells, p53 exists more as a dimer than anything else, and then after DNA damage, her work has shown that it assumes the tetrameric conformation. So we also know that this is also a display of different mutations in p53 with the hotspots. There's this unusual mutation in the oligomerization domain that's known as the Brazil mutation, which I'm not going to talk about today. But as you can see, these patients that are born with one mutant p53 allele tend to also have hotspot mutations. And a very rare mutation happened to be the mutation of this patient that I met at this Lee Fermani conference. It's called A347D, and these are just pictures of the very famous and very wonderful epidemiologists who first described this condition. It was subsequently shown after they found the syndrome that by David Malkin and others that the genetic basis was having a p53 mutation, germline p53 mutation. This is just a display of different kinds of mutations in Lee Fermani, including the dimer forming, very small proportion, but a very, very interesting mutation. So we worked with two different systems to study this. The first are actually derived from Lee Fermani patients, and this was actually a paper that was first published from Ihor Lemishka's group many years ago, and then Dongfang Li in his lab took this with him because he was the postdoc that developed it, and we ended up collaborating with him. Cell lines, fibroblasts from patients are subjected to the Yamanaka dedifferentiation protocol, iPSCs are made from which mesenchymal stem cells are generated, osteoblasts, and he showed that in some cases the cells from the patients could produce osteosarcoma-like proteins. So he did this experiment with the cells from these patients. We had two. Now the patient that approached me had a sibling with the mutation and another sibling without it. And so we work with these cells, but we also generated a set of CRISPR cell lines using U2OS cells. These are osteosarcoma cells that have wild-type p53, and from them were generated heterozygotes, homozygotes, and knockout cells. So this is a great allelic series to be able to work with. The experiments I'm going to describe, basically we did are mostly with U2OS, but almost all, wherever we could, were also confirmed and extended with the patient-derived osteoblasts, but I'm just not going to show that data here. So if this is a dimer-forming mutant, as it was predicted to be from work that was actually published years ago from a Japanese group who did a lot of mutational analysis of p53, does this mutant form dimers? And it certainly does. Basally, it's very overexpressed, and you see a very large amount of the dimer. This is where the tetramer runs over here. If you treat the cells with etoposide, you now get lots of the tetramer, still get some dimer, but you get no tetramer whatsoever in the homozygotes, and just a little tetramer with the heterozygotes. When you look at how these cells work in culture, they can be induced by nutlin, but only slightly if you've got the homozygote. And it seems to be, from this, completely unable to induce its major targets, p21 and Mdm2. So this led us to collaborate with Dongfang Li, who also did RNA-seq analysis with us. And you can see that you can get a huge induction of many different p53 targets, either with the heterozygotes or with wild-type p53. But as soon as you lose any wild-type allele, basically you get a very weak response and very little evidence for any significant induction of genes whatsoever. So we looked at this as sort of a loss of function. You're just losing the ability to activate target genes. However, when a ChIP-seq was done, it was quite interesting. There was a lot of peaks, but they were completely different than the peaks you would see with either wild-type with the wild-type p53. And if you looked at actually the genes that were under these peaks, you saw some very interesting properties, including a lot of, let's say, pathways involved with mitochondrial function in the cells, and I'll come to that in a moment. So in some way, you can call this a gain of function, because you've acquired new binding sites that you don't see when you're clearly not going to see when there's no p53, and you don't see when there's any wild-type p53 present. There's also a transcriptional and metabolic protocol that's unique to these cells. They acquire something of a glycolytic phenotype, which is, I think, very interesting. Also a gain of function. I'm not going to spend any time on that. But the thing that was visually the most striking was that if you put these cells under a microscope, their mitochondria looked really strange, very disorganized and stretched out. And it was completely a function of the homozygotic cells, not either the heterozygotes or the knockout cells. And we know that it wasn't just some aberration of deriving these cells, because if you knock down p53 in them, you lost generally this abnormal morphological phenotype. So this is another gain of function. So I'm giving you some examples where a mutation can actually confer unusual properties on the cells. So I'm arguing that's gain of function. I'll be talking more about that in a moment in another context. But one of the unusual properties of these cells that might stimulate ways of treating patients with, for example, this kind of mutation was their hypersensitivity to topoisomerase II inhibition. And etoposide was shown to be very, very toxic to these cells. And if you could reverse this cell death, where this is actually a viability assay, with ZVAD, indicating the kind of cell death was apoptosis. What was very interesting is, as I told you before, they seem to be transcriptionally inert. So one question the reviewer of the paper asked is, how do you know there's any transcription involved needed at all? So the cells were treated with actinomycin D, and we got the exact same sensitivity. So this is a completely transcription-independent response to a chemotherapeutic agent. So that means this p53 can do something to cells that has nothing to do with its very major function as a transcription factor. So what could that thing be? So it had been reported earlier, first by Utamal and then by Doug Green and others, that p53 can actually bind to mitochondria. And so I'm not going to show you all the data, but we definitely had evidence that you could find p53, this is another gain of function, by the way, associated with mitochondria. So several years ago, a lot of years ago, Andrei Gutkoff's lab reported a protein called p53, which he said could disrupt p53 binding to BCL2 and BCXL, these mitochondrial-associated proteins. And although I won't show it here, we were able to show you could co-immunoprecipitate the mutant with these two mitochondrial apoptotic regulatory proteins. BCL2, of course, protects cells from apoptosis. And so we thought we would test p53. This is actually a very good idea of Josh with this drug. And so first of all, as I said, we could find this particular mutation with mitochondria, and we could show that if you added more p53, it decreased the association with mitochondria. But most strikingly, if you, just these things, if you treated the cells with p53, you actually significantly rescued the cell death caused by etoposide. So this means that the cell death caused by etoposide was actually caused by p53's association somehow with mitochondria. We don't know how, but this is a transcription-independent, mitochondrial-dependent form of cell death that p53 can cause. p53 probably has to be a dimer to do it, but I still think that's a very interesting observation. And so this just sort of summarizes this part of the talk. We believe that this version of dimeric p53 somehow interacts with mitochondria. This may be linked to the altered mitochondrial morphology. I think there's a good bet on that. And the topo2 inhibitor actually causes a mitochondrial cell death that requires p53s associated with the mitochondria. So these are the take-home message is a very stable dimer-forming mutant found in a Li-Fraumeni patient family. It has several gains of functions involved in metabolism and mitochondrial changes. I have to say as well, though, that it didn't increase tumorigenesis. In other words, if you did a xenograft assay, there was really no difference between knocking out p53 and knocking in this mutation in these U2OS cells, which happen to be rather weakly tumorigenic anyways. So, but nevertheless, I think this really opens up the idea that you can have a version of p53 that can do stuff to cells that has nothing to do with transcription. So, moving on. Now, another person in the audience might be very interested in what I'm going to talk about now. This is the work of Will Friedpaster, who, when he was an MD PhD student, I'm sorry, it's a bit sort of squashed, this picture, if you will. You're better looking than that. We actually acquired some cells from Jill Barganetti, which had an inducible shRNA against p53. And Will very courageously set up for the first time in our lab the 3D system that had been pioneered by Mina Bissell and beautiful work done by Joan Brugge in this area, and showed that these cells normally look pretty abnormal in culture instead of what they should look like, which are like little acinar-like structures. And when he actually induced the shRNA, you got far more of these acinar-like structures indicating a phenotypic reversion to a more normal phenotype. So this is a story. I'm over 10 years old. I was very proud of this work that Will did. He really opened my eyes to many different aspects of cell biology. I have to say, if you're in the p53 field, you know a huge amount about p53, but you only learn a little bit about many other fields like DNA damage, signaling, like transcription, like cell biology. I find cell biology particularly intimidating. And the last part of this talk is cell biology, so forgive me. So one of the things Will did was collaborate with other labs that we had a program project with, including Arnie Levine's lab, and did a gene expression analysis and came up with the rather surprising result that a pathway that was very significantly affected when he ablated p53 was the methylonic acid pathway. This is the pathway, as many of you may know, by which cells synthesize not only cholesterol, but several really critical byproducts, such as the isoprenyl moieties that are needed for attachment of small G proteins to membranes, and I'll be coming back to that in a little while. It's also, of course, the pathway that is, the target of these blockbuster statins, as well as inhibitors of different aspects of the pathway, particularly the isoprenylation and other things. It's a very important pathway in cells, and I'll come back to that in a little while. So I'm not going to show you any of the data, but Will developed the hypothesis that mutant p53 is collaborating with this master transcription factor called SREBP2 that's responsible for activating virtually all of the genes in this pathway by a very beautiful kind of regulated pathway, which I'll come to in a little while. And he built, Will, you'll recognize this slide too. The idea was that when mutant p53 is associated with SREBP2, it's increasing the levels of sterile biosynthesis enzymes, which is responsible for this morphological phenotype. And if you have less of mutant p53, you end up with a more normal phenotype. It's much better on that screen than this screen, by the way. So that's the story here. Oh, yes, and then this is sort of, I don't know if it's pre-TCGA, but we certainly didn't have access to it. We collaborated with Anna-Lisa Boris-Dale in Norway, who actually had quite a collection of gene expression analyses of breast cancer patients. And looking at her data, Will realized that there was a really beautiful correlation between higher levels of these mevalonic pathway enzymes and not only p53 mutation, but actually survival of the patients. So patients that had higher levels of these enzymes had significantly less survival than, Annalisa must have had the survival data, than the ones with lower levels. So that's this slightly old story. I'll be coming back to it in a moment. But one of the fascinating things about p53 is that there's a really odd sort of reciprocal relationship between the pathways and the genes that wild-type p53 regulates and the pathways and genes that mutant p53 regulates. So very often, for example, very famous p53 targets that are activated by p53 are actually lower in cells with mutant p53 than if you have no p53. And so and vice versa, targets that are higher in mutant p53 cells are very often lower in wild-type p53 cells. And so having made this connection to the mevalonic genes, a pretty obvious question was to look at the possibility that wild-type p53 is downregulating these genes. And this was really the work of Sung-Won Moon, who was a postdoc in the lab at the time, who did a very good job, and we had a fabulous collaboration with Scott Lowe and members of his lab in that respect. So the idea was, what happens if you turn on wild-type p53? What happens to the mevalonic pathway genes? And this is a horrible little slide here, but basically the genes get expressed less well, and this is using a larger panel of genes, using a very famous, venerable pair of isogenic cell lines, HCT116, from which many years ago the Vogelstein group deleted p53, and you can see a significant decrease in the expression of these mevalonic pathway genes compared to the p53 null cells. And then when the cancer cell, I guess it's the Cancer Cell Encyclopedia was examined much more, almost 500 cell lines, there was a pretty striking inverse relationship between having wild-type p53 and the expression of these genes and having mutant p53. So that was very encouraging. So then came the big question, how is this working? So as I said, because many genes were affected, maybe this was linked somehow to this master transcription factor, SREBP2. And so this is what we know about SREBP2. It's an exquisitely regulated protein. It's in one of a family of 3 that are held as inactive precursors in the ER. But when the cell senses that there is a depletion of cholesterol or other sterols, the SRE precursor is transported to the Golgi, where it's subjected to cleavages, and then it gets into the nucleus and binds to these sites. And so there's this very well-regulated cleavage process. You have a precursor that's inactive and a product that's active. And so one of the first things that Sung-Won found is that you got significantly more of the mature form in the p53 null cells. And he was able to show this not only with these cell lines, but for example, if you turn on p53 with nutlin in a cell line such as SKHAP1, you get less of the mature form. If you turn on p53 sort of artificially in another cell line, you again get a reduction in you get a reduction in the mature form of this protein. So this is like the kind of story that happens over and over in my lab. You end up with an observation, and then you just spend a lot of time trying to figure out how this works. And so Sung-Won must have spent well over a year trying every possible obvious target. And then what he did, and this is sort of an advice for you, is he just started reading the literature a lot. And you read and read, and you finally come up with something. And he discovered a paper from a group from Japan who had made the rather surprising result that a transporter, a lipid transporter, actually a cholesterol transporter called ABCA1, not only functions to export cholesterol from cells, but it can actually, under certain conditions, perform what you call retrograde transport, where it can move cholesterol from the cell surface to the ER. And so he tested that, and it turns out that ABCA1 is very likely the reason for this ability of p53 to repress expression of the mature form of SREBP2. So first of all, you can activate ABCA1, and if you get more ABCA1, you see less of the precursor. And this is just knocking down ABCA1 as a control. And then the other question is ABCA1 a p53 target? And yes, it is. It has a sort of a p53 binding site in the promoter. Not bad, and you could chip p53 on it. We didn't look that carefully to see if there were other sites. But what you can see is that if you induce p53 with, let's say, nutlin, you get a big increase in ABCA1 protein and RNA. And you can do this in another system where you turn on p53 in these 12-9 cells or a transcriptionally defective version of p53, and you'll see ABCA1 going up only with wild-type p53. So while we were doing these experiments, we were also collaborating with mouse models with the Scott Lowe lab. And I'm not going to show you all the data, but they were working using a technology called hydrodynamic tail vein injection, where you inject constructs under high pressure into the tail vein of the mouse. And that ends up with these constructs somehow being produced or released into the liver via the portal vein. And they showed at the time that if you combine an shRNA against p53 and another oncogene, such as MYC, for example, that you'll get liver tumors that have a lot of the characteristics of hepatocellular carcinoma. They also showed, and I'm not showing it here, that if along with this you knock down key mevalonic pathway genes, you got much fewer tumors. So that was a very satisfying result. But when we submitted our paper for publication, we were told, what about ABCA1? And so Shauna Houlihan in the lab now included, I think it was basically a CRISPR guide RNA against, guide construct against ABCA1. And yes, she did get tumors. She did see them. They were obviously, this is what happens when you ablate p53. It's much more earlier and more tumors. However, I think it's safe to say that there is no single p53 target that can completely phenocopy the impact of knocking down p53. So this was actually a pretty good result. In fact, people have knocked down 3 different major targets and not seen anything, or seen nothing like this. So basically, and this has just shown that you could see more of SREBP2 in these knockout cells. So basically, what we had at the time was, this is good, we had from Will's work and from Sungwan's work a really interesting relationship between p53 and the mevalonic acid pathway, where mutant p53 is somehow working with SREBP2 to activate this pathway, and wild-type p53 via ABCA1 is dampening this pathway. And so the next question is, when you introduce mutant p53 into a mouse, as opposed to a knockout, you get metastasis. And so the idea is there a link in this case to metastasis? So I don't have actual metastasis data to show you, but Aja Guzman in the lab has been working on a protocol that allows one to at least measure a very early stage in the metastatic process. And we've also had some very good collaborations on this as well. Aja actually came to my lab from Laura Kaufman's lab. Laura Kaufman is actually a chemistry professor who's very interested in biomaterials, including the kind of matrices that cells can sometimes invade into. So Aja had already sort of established this protocol. She didn't develop it. I mean, she didn't invent it, but she did a lot of beautiful work with it. And the protocol involves taking cells, growing them in 2D, and then dispersing them and putting them into what are known as low adhesion tubes. And if they sit there long enough with the right medium and serum, they actually form a spheroid. And you can control the size of the spheroid, obviously with a number of cells, and so on. And then the spheroids are actually embedded directly into various biomatrices. So collagen is a good one, because that's actually, as I understand it, collagen is a major component of the extracellular environment of some breast tumors. And basically, what you can do is that within 24 hours, the cells will start to invade out of these spheroids into this extracellular matrix, biomatrix, whatever you want. And as it says here, there's about 10 or 12 spheroids per condition. And of course, these experiments are repeated several times, so you do get statistics. So I'm going to show you 3 different examples of what happens. Basically, you need mutant p53 to get out of these spheroids efficiently. So in this case, this is just knocking down p53 with siRNA in the same 468 cells that Will used originally in his experiments. Another cell line, so the thing about 468 cells is that they also need mutant p53 to grow in our hands. So in other words, we can't knock it out of those cells. That's a whole other story. You can knock out p53 from another breast cancer cell line 231. So we have CRISPR clones of these, several of them. And then finally, we were able to knock into HCT116 cells a hotspot mutant R282W that happens to be the most frequent mutated position or codon in colon cancer. So that's very interesting. In each case, If you knock down or knock out p53, you get much less of this invasion out of the spheroids. And conversely, compared to the knockout or the wild type, you get more invasion when you knock in a mutant in these cases. So what do we learn more about this? So first of all, for a number of reasons, we still haven't set up a metastasis assay. We're working on that. But Gizem Efe, who's an amazing member of Anil Rustigi's lab, and we're collaborating a lot with them. You'll see her again in the last part of this talk. There's an assay that she read about and set up, which is basically, it's kind of an ex vivo assay. You take lungs from mice, poor things, and you decellularize them, and then you basically incubate them with whatever constructs you want, and then you wait several days, actually, and you looked for colonies to form. So you're basically looking at for P50, I'm sorry, it's not constructs, it's cells you put on them, not constructs, big mistake. So you put either your wild type, not your wild type, your normal mutant cells or your knockout of these cells on this lung assay, and you just wait a few days and you'll see colonization in this assay. So you're basically looking for invasion through a more, let's say, normal physiological tissue. And in fact, lungs is actually a common target of breast metastases. And you see a big effect here. So going back to the spheroids, so now you can ask interesting questions, and here I move into the scary cell biology part, so bear with me. First of all, it's been known that when, I'm not going to show this data, but when cells invade out of a spheroid, they can either do it as single cells or as what you call collective groups of cells. And both of them have been identified, and collective migration is supposed to be particularly relevant to metastasis. And although I don't show it here, mutant p53 controls both single and collective migration. Also, it maintains the shape of the cells. And so the cells, when they're elongated, actually migrate less well through this particular biomatrix. And mutant p53 maintains a more circular shape of these cells. Now here I really move into an area which I don't know what I'm talking about, but more rounded cells apparently have little structures on them called blebs. And Asha had actually studied blebs in her previous experimental lab, and it turns out that there were far fewer blebs on the cells that she had ablated, that from which didn't have p53. So it's sort of in its simplest form, it's been shown that a rounded cell shape is correlated with having much higher bleb morphology. And what blebs do is they play a role somehow in cytoskeleton contractility, and cytoskeleton contractility is a product of Rho and ROCK signaling. So this led, of course, Asha to ask, where is the role of Rho and ROCK in this? And it turns out that if you block either rosin, which inhibits Rho, or Y27632, which inhibits ROCK, in either case, you get a big drop in invasion. And if you knock down mutant p53, you don't get further drop. So this implicates mutant p53 in Rho-ROCK signaling. So Rho-ROCK signaling, in turn, requires basically attachment to the membranes through isoprenyl moieties. And so we circle back now to the mevalonic acid pathway. And it turned out that if you inhibit this pathway, either with a statin or a drug called phaedostatin, which inhibits the maturation of SREBP2, or a geranylation, in each case you get a big block, a drop that's pretty similar to the reduction in invasion when you see with mutant p53. And this can be completely reversed by supplying the cells with geranylgeranol pyrophosphate. So this implicates geranylation in the ability of mutant p53 to invade out of these spheroids. So Another way of asking is, does the association of RhoA with the membrane, which is what the isoprenylation is needed for, affected by mutant p53? And this is with mutant p53, and this is without it. And so this is actually a very interesting technology where they basically do cryosections of these spheroids, where they're doing metabolomics on them as well. But you can look at single cells and see differences using this approach. And if you do a classical fractionation, you see a big drop in RhoA associated with the membrane when you ablate p53 with siRNA. So in the simplest way we can develop a hypothesis is that the mevalonic acid pathway is actively repressed by p53. Therefore, you're going to get more expression with loss of p53, and hopefully even more, well, hopefully as a scientist, but not as a person, you'll get even more with mutant p53. And we have some evidence of that. I think the one that I find most satisfying is we know that if you knock down p53 in these spheroids, you get less expression using basically RT-PCR and looking at these different genes. But with some of these genes, where we knocked in the mutant, the R282W, you see significantly more, not all of them, but some of them, when you actually have the mutant present than when you either don't have p53 or you have wild-type p53. So we focused on IDI1 for a number of reasons. And we looked in the TCGA, and like we saw in our experimental system, the levels of IDI1 are significantly more in each case when you have a missense mutant than when you have a truncation mutant, which in turn is more than when you have wild-type p53. And there's a small impact on survival as well. And finally, the same wonderful person who collaborated with Josh, who also collaborated with Aja, he generated an ID1 overexpressing cell line, and sure enough, it's much more invasive than the control. So basically, our flow is in mutant p53 is needed for upregulating the mevalonic pathway genes, very likely through some way of interacting with SRABP2, which is needed for the attachment of RhoA to the membrane, to ROX signaling, and cell migration. That's where we are with that. We hope to have some metastasis data soon. So the third part moves into a very different way we're thinking about this whole pathway in p53 and MDM2, which is the possibility, and this will be the last part of my talk, that MDM2 hasn't just evolved or doesn't just function as an obligate regulator or repressor of p53. It has a life of its own. And in fact, we're certainly not the first people to have discovered this. I should say this work was spearheaded largely by Rafa Muniz. She's a wonderful postdoc in the lab. Aja collaborated with her, as did Gizem Efsani, the person I described before, and we had a very good proteomics collaboration with colleagues in Japan. And just to remind you, MDM2 and MDMX are these two proteins. They have a lot in common. They both have p53 binding regions, acidic domains, and ring or ring-like domains. And this is, and the interaction between these two is actually inhibited by a drug that was developed in Brent Stockwell's lab called MEL23. It inhibits the complex not from forming, but from acting. And it inhibits MDM2's E3 ligase only when it's in complex with MDMX. So, and so finally, during the first stage of the pandemic, when we weren't allowed to go to work, 3 lab members got together and decided to write a review on all the different ways in which MDM2 can do things in cells that don't have p53. And believe it or not, there's a lot out there. It's been shown to be associated with tumorigenesis, with metabolism, with chromatin association, and so on. There's actually quite a lot of things that you can watch or observe MDM2 functions that you can observe it to have when you don't have p53. It's very hard to really, I guess, picture this because our cells all have wild-type p53, but many tumors don't. And so it's an interesting question, whether you have no p53 or mutant p53, how is MDM2 functioning? It's definitely functioning in vitro in cell culture assays. Whether it's functioning in real tumors, we don't know, but I hope we find out someday. What we learned in our cells was that if you, and we had other people have shown this, was that if you deplete MDM2 with siRNA, or in this case, this is a cell line in which we established an shRNA against MDM2, you get significantly less migration in a classical wound scratch assay. You can also set this up in this spheroid assay that I described before, and again, knocking down MDM2 or inhibiting with MEL23, you get a significant reduction in invasion into the extracellular collagen matrix that spheroids are embedded in. In fact, here we were able to do a metastasis assay, or more accurately, Gizem and Anil Rustigi's lab did. And very strikingly, 2 different metastasis assays show a huge drop in metastasis when you ablate MDM2. In this case, these are stable shRNA expressing cell lines. I should say there are some cell lines that require MDM2 actually for growth and survival, but others don't. And this particular cell line, HT1080, is very minimally affected by ablating or ablating MDM2. So this is an orthotopic base assay where you inject the construct into the the femoris of the mice, and then eventually a tumor forms and then the cells are sacrificed. Or you can inject directly into the tail vein. It's a classic assay. I just lost my image here, which is not good. Whoops. Okay. What happened? How do I get it back? It's terrifying. It's back. I know. Okay. You can still see stuff there. Okay, that's fine. It's fine. Okay. It's a little weird here, but it's okay. So, so we see much less lung metastases. So we also saw, after a lot of work showing that MDM2 is needed for basically the cell's ability to put down ECM onto the plates. Does this thing work? Yes. Rafa also discovered that focal adhesions, these little structures that sort of connect the cytoskeleton to the extracellular matrix, were greatly reduced in these cells. And then, of course, like as I said in the description of the earlier project, she spent a huge amount of time trying to figure out what's going on. And finally, we decided to, this is just what a focal adhesion looks like. We decided to do a proteomics experiment with our colleagues in Japan. This is an unbiased, non-targeted proteomics experiment where you simply look at the proteome in a cell that's been treated either with 2 siRNAs, cells that have been treated with either 1 siRNA or the other, or MEL23, the ones we'd used in our experiments. And I was really shocked by these data, because in common with all of these were only 8 targets. And in fact, if you could narrow that down to 3 that went up when MDM2 was inhibited. So look at all the basically the off-target effects that you see in a cell when you treat it with anything. So you have to keep that in mind when you treat cells with stuff. A lot of other things are happening in cells. So nevertheless, Rafa soldiered on and chose one of them, of the 3. She actually tested all 3. One of them actually gave her the answer. And interestingly, it was a gene and a protein called SPRODY4. SPRODY4 is both a target of the receptor tyrosine kinase signaling and also actually an inhibitor. It feeds back. It's been shown to negatively regulate migration, been shown to be down-regulated in some cancers. This is just another diagram of that. And she was able to confirm that, in fact, if you downregulate or inhibit MDM2, the levels of SPROD4 go up quite a bit, maybe one and a half to twofold, not dramatically, but quite a bit. Interestingly, the regulation is at the level of RNA, not protein. And interestingly as well, if you co-deplete SPROD4 and MDM2, you get a complete rescue of both migration, the formation of focal adhesions, and very dramatically of metastasis. So we think that we've identified a new player, I guess, in the relationship between MDM2 and processes in cells that are related to metastasis, which of course is the most deadly and one of the most important ways to think about cancer. So I've just finished with, oh, I won't finish. I'm just going to finish here. Thank you very much. I was going to, thank you. Yeah. I just wanted to say, I actually should have had a slide there, that there's a lot of drugs have been developed to go after MDM2, and there's people here doing beautiful work on them. And I think we have to think about some of these drugs in the context of cells that don't have wild-type p53 and try to understand how they work. So thank you. That's one good way to get two rounds of applause.
Any questions?